Hi, everyone. My name is Christine Marshall, and I'm an instructor in biology here at Phillips Academy. And tonight it is my great pleasure to introduce Dr. Don Ganem, class of 68. Um, as you can imagine, it is such a joy to work and teach at Andover. One of the best parts is the students, right? They're amazing people. Um, you were all amazing when you were here. Parents, your kids, we love so much. We love to work with them. Another great aspect of this job is meeting these students once they go out into the world and become incredible alums. And Don has been incredibly generous with us and has visited to campus frequently. He's come to talk to our bio kids and guide their thinking about careers and entice them with some tales from his research. Don has really had two careers. I don't know, Don, how you've done it all, but he has had a very successful career in medicine. He is a virologist by training and a medical doctor. He's worked very hard to understand the mechanisms of the hepatitis B virus and the Kaposi sarcoma associated herpes virus, and worked to understand numerous other viruses in humans throughout his career. He notably trained with Harold Varmus, who's a Nobel laureate, and then Harold convinced him to stay on the faculty at UCSF and become a professor himself of infectious disease and medicine. And after Don worked to understand a number of diseases and really understand the new viruses that were emerging, he left academia for about 7 or 8 years to head a global research team to, with the goal of developing new antiviral medicines, particularly for hepatitis B virus and other human herpes viruses. And to develop antibiotics for multi-drug resistant bacteria. So it's very unlikely, it's very unusual for a medical doctor to be able to not only understand new mechanisms, but then to also have a full career developing treatments to cure some of the diseases that one has spent their time working on. Don is now back at UCSF as a professor emeritus. He has held numerous positions. He's a member of the National Academy of Sciences, the National Academy of Medicine. He was a Howard Hughes investigator, a very prestigious post, and also president of the American Society for Virology. So I know that you all have a lot of questions for Don, and we're going to walk through a program tonight, and we've been cataloging the questions that you've been sending in, and we are going to aim to reserve some time for Q&A at the end, and we'll do our very best to answer any questions that you've sent in or that come to mind in the next 45 minutes or so. So I wonder, Don, whether we might start by looking at some of the different types of treatments that are out there, ones that folks maybe are hearing about in the news. We have one medicine that's received a lot of press, remdesivir, which is a drug that attacks the virus directly, preventing it from operating and spreading through a person's body. And you and I talked a bit about the clinical trial that has gotten a lot of press and why it may have given some optimistic, but maybe less impressive results than were originally anticipated. So could you explain the timing required for the drug's efficacy, maybe when and how it could be employed in the future to limit the number of COVID cases that we're seeing in the hospitals?
Sure. So first of all, let me just say it's a great pleasure to be back. Electronically at Andover, if not in person. It'd be a lot more fun to be there in person. It'd be great to do this with a live audience. But anyway, this is our COVID time. So I'm just so happy to do this. And so let's get right to your question. I think before we talk about the drugs, we need to say just a couple of words about the natural history of infection. What goes on once you get exposed to the virus? and how does it progress through the body. So once you get exposed, somebody coughs or sneezes on you, coughs on you, and you inhale the virus into your upper airway. There's replication in your upper airway, in the back of your nose and your throat. If I slip into medical parlance, that's called the nasopharynx. So nasopharyngeal replication, upper airway replication in the nose and throat, especially in the deep recesses of the nose, in the back of the throat. That's where virus replication begins, and the virus gains a foothold in the body there. And then it spreads down the respiratory tract, down the trachea, into the bronchi, and into the small airways of the lungs. Now, in the first few days of infection, most of the infection is confined to the upper airway. And interestingly, a lot of people don't have a lot of symptoms right then. A runny nose, which is very common in other respiratory infections, is relatively uncommon here. It can happen. Congestion, stuffy nose, even overt runny nose and sneezing can happen, but they're much less common here than they are in the common cold. Okay? But meanwhile, replication is going on and viral titers are rising. The levels of virus, or the viral load, is rising in the upper airway, in your nose and throat. And then as days go by, day 5 or 6, there's 3, 4, 5, 6, there's spread to the lower airways in a lot of patients. And when the virus spreads into the lungs, that's what we know as pneumonia. Initially can be mild, but, and there's a lot of variability here. But as time progresses, by the time you get to day 7 or 8, levels of virus in the upper airway and in the respiratory secretions start to go down, such that by day 8 or 9, levels are usually quite low. It's very difficult to culture virus from respiratory secretions after day 8 or 9 or 10, even though we can still detect viral RNA at lower titers. But so the general impression is that the early part of the infection is where there's a lot of replication. Later on, after spread goes into the lower airway, the total amount of replication seems to go down. Now, it probably doesn't go to 0, but it's certainly going down. And then you start to get into the second phase of the illness. Now, in some people, it just ends right there. There's an immune response, you clear the virus, and people get better. That happens to a lot of people. They just plain get better. But in some people, the viral replication in the lower airway kicks off this very aggressive inflammatory and immune response that we don't understand very well. But when that happens, it causes the lungs to fill up with fluid. Even if there's not that much virus around anymore, you're now talking about a phase of the infection where the immune response to the pathogen is starting to exceed, the damage caused by the immune response to the pathogen starts to exceed that caused by the pathogen alone. And in other words, it's kind of a biphasic illness. Early on, lots of replication in the upper airway, some replication in the lower airway, ongoing immune response. That's enough to clear the infection in lots of people, but in a fraction of people, it's too aggressive and triggers this unrestrained inflammatory and immune response that gives rise to a lot of tissue damage in the lung that isn't caused by the virus alone, but was triggered by it, caused by immune responses directed against things in the lung we don't understand very well. So this part of it is poorly understood. I'm getting into the hand waving part now. But what's important for lay people to understand is it's a biphasic illness. Replication happens early. The immune response kicks in. That's enough to control the infection in most people, but in some people, if this exaggerated immune response and late in the disease, that triggers a lot of lung injury, fluid accumulation in the lungs, difficulty oxygenating the blood, and that's what gives rise to the hospitalizations, the ICU transfers, and the mortality, okay? And the inflection point in there is somewhere between day 7 and 10, okay? Before that, it's a lot of replication. After that is a lot of inflammation and injury, okay? Now, so when we think about drugs for this, when we're talking about antiviral drugs, drugs that are there to block viral replication, you would expect them to be most useful really early in infection, day 1, 2, 3, 4 of symptoms. But that's a time when people are usually at home and they're not sick enough to come into the hospital. They often don't seek medical attention. And you also have to be aware that not everybody has symptoms. And a lot's been talked about the number of asymptomatic people. So you have a lot of people have lots of replication in the upper airway, have no symptoms at all. Other people with minor illnesses think it's a common cold, or a more significant illness think it's the flu, but they're not bad enough to go to the hospital and just toughing it out at home. But that's the time in this illness where we would anticipate that antiviral drugs would have the greatest effect. Now, remdesivir is a very interesting drug. It was developed by a company called Gilead Sciences in the Bay Area. They were initially trying to make drugs for other RNA viruses, influenza, and other drugs, other viruses that are more common in our population. It didn't work for any of those. It was active in culture against Ebola, but didn't work very well in Ebola patients. But it was active in culture against the coronaviruses, so they decided to try it. Now, the thing about remdesivir is that it is not orally active. It's not absorbed from the GI tract, okay? So you have to give it directly into the bloodstream or intravenously. That means you can't give this drug to somebody who's early in the illness at home when we would think it would be most active. So you have to have an intravenous line to get the drug. Therefore, you have to be in the hospital. So in the clinical trial that came out recently, remdesivir was given to people who were hospitalized and compared to placebo. Okay? And the good news is that even though replication is on the wane then, there's obviously still enough replication going on to see the effect of the drug. People on the drug had hospital stays that were on average 4 or so days shorter than people who were not on the drug. When you look at mortality, it went from 15% to 11%. That drop was not statistically significant in that cohort. The cohort wasn't big enough to see a statistically meaningful impact on mortality, but it was certainly a trend in the right direction. And so that's what we know about remdesivir. And on the basis of that, a statistically significant shortening of the illness and a trend, but not significant improvement in mortality. The drug was approved by the FDA. So that's what we know about remdesivir. Now, we're in a stage where a lot of people are going to be treated with remdesivir. It's going to become the standard of care for everybody admitted to the hospital. That means ethically that new trials of new drugs are not going to be compared against the placebo, at least in hospitalized patients. They're going to be compared to remdesivir. So for ethical reasons, people will get either remdesivir alone or remdesivir plus the new drug in new clinical trials. So that's where we stand with remdesivir. Now, the problem with remdesivir is you can't take it as a pill. It's not orally active. So a lot of pharma companies, biotech firms, and academic labs are screening now for other compounds that might be available in pill form that would be active as antiviral drugs. Interestingly, hydroxychloroquine is weakly active as an antiviral. It blocks viral entry. That was how it got on everybody's radar screen as a potential treatment. But it turns out the clinical trials of hydroxychloroquine have been very underwhelming. They've produced really no major impact on viral replication or symptoms. And they have toxicities, cardiovascular toxicities. They can produce cardiac arrhythmias in susceptible people. So the bloom is off the hydroxychloroquine rose, and I think the FDA has just recently taken it off the list of drugs that it would approve for emergency use. And I'm sure the star of hydroxychloroquine will fade. Although there are still some ongoing clinical trials that are out there, it has proven effective neither in therapy nor in prevention so far. The trials are all a little suboptimal. They're all a little underpowered. I don't think we can say that the final chapter has been written there, but that little musical is winding down.
So if we did go to extensive testing and folks who are asymptomatic or mildly symptomatic test positive, what are the chances that they would be entered into some IV protocol that they could do at home or as an outpatient to prophylactically keep the disease from becoming deep lung pneumonia?
Yeah, I think it's not likely that a lot of outpatients are going to get treated with an intravenous medication. Okay. I'm sure that a few people would be willing to get an intravenous line and take remdesivir, but I think if you consider the global need here and the massive scale of this pandemic, there isn't enough remdesivir right now as a global supply to meet that need. I think the company is really working very hard to ramp up production to meet the hospitalized patient need. And that will have to be satisfied before we can even think about giving it to milder patients, especially since it's going to become the standard of care. But the good news is that there are a lot of trials ongoing. Now, there are academic groups and biotech companies and charities like the Gates Foundation that are screening all the known FDA-approved drugs to see, do any of them by chance impair replication of SARS-CoV-2, the virus of COVID. And there have been a bunch of hits. There are a bunch of candidate compounds that these are drugs that are already either licensed for other purposes, or one of the things that the Gates Foundation orchestrated and a group in Southern California orchestrated was they went around to pharmaceutical companies and said, can you give us all the compounds that you were developing for other indications, cancer, autoimmunity, that made it into clinical trials and therefore for which we have safety data. Even if they didn't prove to be successful for the disease you were developing them for.
Right.
And let us see if any of them by chance happen to hit the SARS coronavirus. Because these are drugs, even though they're not already approved by the FDA, they've been in human beings. We understand what their toxicology is. We know they can be absorbed. So there are a bunch of orally bioavailable compounds now. They're not drugs yet, but they are close to being drugs. And some of them have been pretty active in laboratory experiments. So those are now being, rushed into clinical trials. So in the next few months, we're going to learn a great deal more about the results of all that. Now, be prepared for a lot of disappointments. The whole world of drug development is about disappointment. For everything that enters every 10 compounds that enter a clinical trial, only one comes out the other end. So a lot of these things, like hydroxychloroquine, that look nice in laboratory experiments, are going to fail for one reason or another in a human being, even if they're safe. But some of them may make it into the end zone. And even if they're only partially efficacious, just think for a minute about what the impact of that would be. If you knew that there was a drug out there that, even if it wasn't a total cure, could convert a rapidly fatal illness into something that reduces the likelihood of hospitalization twofold, fourfold, sixfold, even if it wasn't a total cure, but a partial success like that would go a long way to removing the great societal fear of this infection. And with that, the fear that if you went to the barbershop and caught the infection, you'd be more likely to go to the barbershop, more likely to go to your workplace, more likely to go to your kid's graduation if you thought the likelihood was that you wouldn't have a fatal outcome, right? So even a partially effective drug could have a huge societal impact here. That's why I don't poo-poo this kind of exercise very much. This kind of drug development, which is called repurposing, you take a drug that was built for one purpose, you test it for another purpose. Drug repurposing has not been a gigantic success over the last 10 or 20 years. But even a partial success in an infection like this, that is rapidly progressive, potentially lethal, and hugely prevalent, would have a giant societal impact.
That's fantastic. Thank you. Now, of course, we do have a range of treatments that doctors are using to mitigate the symptoms caused by COVID-2 infections. And one that we've heard a lot about recently is the old, very popular steroid dexamethasone. And so once patients have that deep lung infection, it does seem, or at least the news is the news outlets are making a lot about its recent successes. Could you speak to that and why inflammation is so dangerous, particularly in lung tissue?
Right. So let's see. So dexamethasone is a very old drug. It's been around for 50 years. It's a very potent version of corticosteroids, of glucocorticoids like cortisone. It's a much more potent version of cortisone. It's very well absorbed orally. It's extremely potent. It's been used for lots and lots of other things in medicine, autoimmune diseases, it's been used a lot in neurosurgery to control swelling in the brain. It's used for a lot of autoimmune diseases. In fact, it's a very potent drug. It's not the kind of drug people are sent home on. It's usually used in hospitals because it's much more potent than typical cortisone or cortisol, which is what people are sent home with orally. But bottom line is it's very potent, very well absorbed, and it's a very broad-acting anti-inflammatory drug. It causes lysis of a lot of T cells, which is major immune cells in the body. It blocks a lot of cell-mediated immune reactions. It lowers a lot of immune hormones or cytokines. So it's a very potent, broadly acting compound. Now, it has a lot of side effects. It can induce diabetes. It can produce psychotic episodes and behavioral changes at high doses. It can lead to fluid retention. There are a lot of other side effects of dexamethasone. So it's not a drug you toy with, but it's a drug clinicians know how to use. It's very potent and very active, and it's a broadly active anti-inflammatory. So it was one of the first things that people turned to when they saw this uncontrolled inflammatory response going on in the later stages of some patients with the disease. So when people got very sick in the hospital, were on ventilators, headed for requiring a ventilator. This is a drug that people were using in an uncontrolled fashion because they thought it might do some good. The good news is that there's recently, now what's interesting is that uncontrolled use of the drug didn't convince a lot of doctors that it was working because a lot of patients were still dying. You really needed a controlled clinical trial where some patients got the drug and some patients didn't. Patients who were matched for equal severity got the drug or didn't get the drug. And then you look at what happened. Now, the raw data hasn't been published yet, but the press releases are all indicating that there was a substantial mortality benefit in patients who got the drug. Something like on the order of 20 or 30% fewer deaths in the group that got the drug compared to not getting the drug. We're all waiting to see the actual data because these are complicated trials, and the design of this particular trial was particularly complicated. It was a trial done in Europe, in England, but by a very reputable group at Oxford, a very credible group. So the early news is that it seems to be beneficial. If that's all confirmed when this paper is ultimately published, it surely will become part of the standard of care if all of that bears scrutiny. But that's where we are right now, and it's very hopeful. But right now, it was a very complicated clinical trial design. I'm grotesquely oversimplifying it by what I just said. But that's the concept here. And we're waiting to see, is it really all it was cracked up to be? But if true, that would be great news. If also, people need to be aware, been a lot of other improvements in the way we manage patients in the hospital, in their ICU care, the way we ventilate them, the pressures that we use to push air into their lungs have been reduced. The frequency with which we deliver breaths have been played with. People have discovered that ventilating people when they're on their stomach was a particularly useful way to enhance the efficiency of oxygenating people. It turns out when you're lying on your stomach, parts of the lung that are less severely affected can be ventilated better, and oxygen exchange can be improved. So there are a lot of little tricks that ICU docs have been able to learn in the course of this that have improved the management of people in the hospital and had small effects on lowering the mortality rate. Those plus effective drugs like dexamethasone hopefully will continue to improve patients' care. So there's been incremental improvement, but these are all improvements at the margins. These are base hits. They're not home runs yet.
So I wonder, on that note, whether I could pivot our conversation to some discussion about vaccines, because this is something
before we do that, 2 minutes about other drugs that are being tried now. When you look at that inflammatory state that people are in, there are dozens of inflammatory molecules that are up in the blood and in the lungs of these patients.
Yep.
And it turns out we have drugs that impair one or another of each of those cytokines. So there's a cytokine called interleukin-6 or IL-6, and there are monoclonal antibodies to IL-6 that are very effective in some forms of inflammation. So those are being tried. And then there are some small molecules, and there are other anti-cytokine antibodies, anti-IL-1, anti-IL-2, anti-IL-6. They're all being tried one by one, and sometimes in combination. There are also oral drugs that were designed for other autoimmune diseases and that are being used now for rheumatoid arthritis and inflammatory bowel disease that block the formation of cytokines. For the physicians in the audience, these are JAK inhibitors, kinase inhibitors that impair the production of a whole bunch of cytokines. Those oral drugs are now also being used. And there's a lot of optimism going into those trials, but it's too soon to say whether they work. But the point is that dexamethasone is not the only arrow in the quiver. There are lots of other compounds that are being tried right now in a clinical trial. Now we can move on to vaccines.
Okay, thank you. That's really good news. I'm glad you added that. So for those of you who are not doctors in the audience, as you know, vaccines give our immune systems a mini prep course in a particular type of virus so that our bodies are ready to quickly identify a virus if we're infected with it, and are more ready to fight it off. So I wonder, Don, whether you could tell us about what more we still need to learn about SARS-CoV-2 in order to make an effective, the most effective vaccine.
Okay. So the most important question you have to ask about any infection before you start talking about vaccine is this. There's one question that matters here, and that is, does the natural infection that you acquire spontaneously confer immunity to a recurrence, okay, to another exposure? Because the track record over the 70 or 80 years we've been doing vaccination is this. If the natural infection confers immunity, we usually can make a vaccine. If the natural infection doesn't confer infection immunity, that turns out to be very difficult to do. So let me give you some examples. Polio, the natural infection confers very strong lifelong immunity, and we've had very good success making a vaccine for polio. But for diseases like malaria or gonorrhea or HIV, where the natural infection doesn't confer immunity and where recurrences or superinfections are frequent, or where, in the case of HIV, once you get infected, spontaneous recovery from that is almost unknown. In those cases, vaccine development has been extraordinarily difficult, okay? So the first question you really want to know is, does infection, if people get over a COVID infection, are they naturally immune to another exposure? And of course, this epidemic is too young to know that yet from just clinical experience. And the experience with other coronaviruses. So the coronavirus is a big family of viruses, and there are many coronaviruses that just produce the common cold. They're in a different genetic family from this group, SARS-CoV-2, that produced SARS, MERS, and now COVID. But so in this other family of more trivial coronaviruses, the experience has not been encouraging. A regular infection by a common cold coronavirus, there are old experiments from the 60s where people were given a common cold deliberately. They recovered from that. And then they were challenged a couple of months later with a common cold virus and got another cold, okay? So that experience with these more distantly related coronaviruses was very discouraging to me. However, there's good news on the horizon. A friend of mine who's a professor at Harvard named Dan Baruch has done an experiment. SARS-CoV-2, the COVID virus, can be transmitted to monkeys, to rhesus monkeys. And rhesus monkeys are primates. They're much closer to humans than most other animals. In fact, SARS-CoV-2 can be transmitted to a lot of mammalian species. It originated in bats. It grows pretty well in cats. It can be transmitted infrequently to dogs. There have been natural examples of cats, big cats like tigers and leopards getting infected. So it can infect a lot of mammalian species, and it infects monkeys, rhesus monkeys. Rhesus monkeys get a very mild disease. It doesn't reproduce the full human disease, but they get an upper respiratory infection. They get a fever, they get a bit of a cough, and then they get better. Okay, and when they get better, and during that time, you can recover virus. There's active replication in the upper airway. There's replication in the lungs. Then they get better, and once they get better, they make neutralizing antibody, and just like we do. Okay, so even though it's a milder infection in rhesus monkeys, it otherwise looks very similar, primarily respiratory, a little bit of infection of the GI tract, then they get better. So Dan has done an experiment that was published a couple of weeks ago in Science, in which he infected deliberately a group of rhesus monkeys with several different doses, and they all got sick and they all got better. And then a month or two later, he challenged them with varying doses of the same virus, this human SARS-CoV-2. And what he discovered was that none of the animals that were challenged got sick at all, but they did get infected, but at a very, very low level, 1,000 to 10,000 fold. When you do quantitative PCR to measure the viral RNA in the upper airway, the levels of viral RNA in those animals was 1,000 to 10,000 times lower than in the naive animals, okay? So in other words, there was what looked like a clinically very significant immune response. They cleared it, they had a low-grade infection, they cleared it much more rapidly, they boosted their titers of antibodies, and they did not get sick. So this is what's known in our trade as non-sterilizing immunity. The animals did get a low-level infection, but they cleared it very rapidly and they did not get sick. Now, these are just monkeys. They're not humans, but they are the closest thing to a human that we can do an experiment on. And so I regard that as extremely promising. And in fact, in a second paper, Dan did another experiment, which is instead of inoculating the monkeys first with live SARS-CoV-2, he gave them one of the prototype vaccines, a DNA vaccine. That's one of the 100 vaccines that's currently being tried. That DNA vaccine behaved in the monkeys as it was expected to. It led to antigen production and neutralizing antibody responses. The neutralizing antibody responses given that those monkeys got were just as potent as the antibody responses that the other monkeys got who were exposed to live SARS-CoV-2. And when he challenged those monkeys who'd gotten the vaccine with live virus, they had non-sterilizing immunity just like the monkeys that had recovered. So this pair of papers, on the one hand, yes, they're in monkeys, but they both showed that sterilizing immunity, non-sterilizing immunity is possible. It's possible with a natural infection, and it's possible with a vaccine. So I regard that as tremendously hopeful news. Okay, and
can I ask a question?
Yeah.
So were some of the monkeys low? You mentioned that some of them had low levels of infection. Were they asymptomatic, and did they also carry immunity?
They were asymptomatic, and then they cleared the infection and had solid neutralizing antibody responses. Now, we haven't re-challenged them again, so the good news is that a primate species that is not too far from humans shows that naturally occurring infections do confer some level of immunity, non-sterilizing but substantial immunity, and that immunity can be mimicked by vaccination. Now, there's one important thing that this experiment doesn't establish, which is the animals were challenged like a month or two after they got over their original infection. That's at a point when they have very high levels of immunity. How long does that immunity last? If you were to challenge them a year from now, would they be resistant to infection? We don't yet know the answer to that. And when I asked Dan on the phone the other day whether he was going to do that experiment, he said no, because they can't afford the animal costs of keeping primates in captivity for a year before they re-challenge them. So, but the good news is there are clinical studies being launched now. We're in the middle of a global pandemic. We have lots of patients who've gotten over a natural infection and have neutralizing antibody. We need to see what happens when those people go back out in the community and get re-exposed, okay? And I've been advocating publicly for experiments to be conducted in these meatpacking plants, on these Navy ships that, like these aircraft carriers where half the crew's been infected. Those sailors, they still are in the Navy. They're recovered, they have antibody, they're going back on those ships. Those ships are going to put into other ports where SARS-CoV-2 is spreading. This is an opportunity to learn, does the natural human infection confer immunity? And we really need to do that experiment. We need to be doing it now. And I think this kind of experiment is underway, but we need to go to places where people are being highly exposed. Healthcare workers in the Bronx, sailors on ships. I mean, there won't be any tourists on cruise ships for a while, but workers in meatpacking plants. These are people from whom we can learn a great deal. The trouble is, I'm sure the meatpacking industry isn't interested in calling attention to the high level of infection in those plants. And so doing an experiment in a meatpacking plant might be difficult. But there are ethical ways to do these experiments, and this is the kind of clinical investigation that we need right now. But the monkey experiment's really, really promising.
So when the other day, when we were preparing for this webinar, we were discussing some particulars about transmission. And I know that transmission is something on everyone's mind because we are starting to open back up. And as folks go back to the stores more frequently and start to convene in greater numbers, you were talking a lot about modes of transmission and reducing the risk. And so I wonder, you sent me some clips for 2 short videos that show an interesting experiment using masks. And I wonder if we could have time to go through it now. I know I'm mindful that we're at 7:41, but I think it was a powerful video to show the impact of masks. And it is a contentious issue, and there's so many folks that don't feel the need to wear them. But perhaps we could show the 2 short clips using my screen sharing, and then we could talk about practical aspects of assessing risk and mitigating risk.
Yeah. Okay. While you load those videos, I'll be talking while you do it, just to
okay. Sounds good.
Introduce. Everybody knows that the main route by which this virus is spread is droplet spread from respiratory droplets that are loaded with virus. And those droplets come in a continuum of sizes, little and big. And let's watch the video now, because visualizing those droplets has been difficult, but there have been advances, and I want to show you some.
Okay, great. Now, I know when I was watching this, I needed to make my screen very bright.
Everyone knows that when you sneeze, tiny saliva droplets spray over a large area. And if they contain virus particles, they can make others sick. But how can people without symptoms that do not cough or sneeze transmit COVID-19? We spew out tiny droplets that drift around in the air and can be inhaled by people near us. If those droplets contain coronavirus particles, their next breath could trigger a new COVID-19 infection. These airborne droplets are fleeting and difficult to see. To visualize them, we routed a high-power green laser through optics that create a sheet of light that passes through slots cut into the sides of a cardboard box whose interior was painted black. With HEPA-filtered air blowing down over the laser system and the box, dust is eliminated, and the laser beam, though very intense, becomes difficult to see. However, when sticking a hand into the sheet of light, the hand and the interior of the box lights up brightly. As a control experiment, spritzing water droplets from a spray bottle over an opening on the top of the box produced a large number of bright light flashes. When positioning one's mouth above this opening and breathing normally, we don't see any flashes of light inside the box. See what happens when speaking the words,
safe, not safe?
The flashes of light you just witnessed arose from droplets of saliva spewing out of the speaker's mouth. Light flashes also occur when speaking other well-intentioned phrases.
Speech droplets can spread COVID-19. Speak through your smartphone, not face-to-face. Thwarting the epidemic through silence.
Okay. That one was a nice introduction. Now I will go to our second.
So this is to show you the impact of mask wearing on the distribution of those particles.
Now I'm recording. Stay healthy.
Great.
Stay healthy.
Great. Less loud.
Stay healthy.
Good.
Now with the mask.
Are you recording?
Yeah.
Stay healthy.
Louder.
Stay healthy.
Louder.
Stay healthy.
Nothing.
Okay. I hope you could all see that. It's a pretty dramatic demonstration of the impact of mask wearing. Now, realize that this is not an experiment about the transmission of SARS-CoV-2, but it does show you two important things. That just phonating, just speaking, generates this droplet cascade. And the louder you speak, the more of those droplets are generated and the farther they are projected. This is why we've always recognized that things like singing, some of you may have read about this super spreading event that happened at a choir practice, a religious choir somewhere in the South. 60 people in the choir, something like 30% of them got infected within days of returning from this event because they were all projecting their voices very loudly. So you can see how attending a mass rally or a demonstration where people are shouting, a political event where people are yelling, and particularly an event where tear gas or pepper spray is being used to promote upper airway secretion, and people are crowded together and all exposed at the same time to a tearing agent. These are tremendously risky events, and mask wearing, of course, should help mitigate a lot of that. But even mask wearing can be overcome by very powerful stimuli to droplet production. So, but I wanted to show you this because, when I watch TV news at night, cable news at night, there's a certain network where people like to get on and say, there's no scientific evidence that masks do anything. And you should be aware of the fact that it's a half-truth. Like all half-truths, it's half-true. We don't have a lot of experiments in which we can show that SARS-CoV-2 transmission was interrupted by a mask. But we just showed you that droplet spread is very strongly interrupted by a mask. And the presumption that there's going to be a significant impact on spread from that experiment is large. And there's now been some epidemiologic experiments, including one published recently, that showed that a reduction in incidence of new cases of COVID simultaneous with the widespread promulgation of masking. So everything we know is consistent with the idea that masks are effective. They're not 100%, they're not perfect, but I just showed you there's a decent scientific basis for the policy, the public policy of advocating mask wearing.
So I wonder if you could help us by describing ways that we as individuals can each do a bit of our own quantitative risk assessment or risk analysis to help calibrate our own risk and guide our behaviors. Certainly masks seem like a no-brainer. It's hard to believe there's so much fuss about them. But this is something that you do all the time in your daily life. You mentioned your wife is also an infectious disease doc and is seeing patients. And so you have a lot of interactions. San Francisco is opening back up. You're at an age that makes you vulnerable. And so based on age and exposure, what do you do every day to mitigate your risk of infection? What are the things you consider, and what are importantly, the actions that you take in response to those considerations?
Yeah. So first of all, just in a general way, people need to understand that risk, you have to make a sort of semi-quantitative analysis of risk. Risk is a function of several things. It's a function of how much virus the person you're exposed to is producing, which you can't know, okay? How close you are to that person, and how long you are in that contact with that person. And you see a lot of apps that are being made by engineers who don't know a lot about biology that are designed to beep every time you come within less than 6 feet of somebody. I don't find that very helpful, because you can't go to a supermarket and in an aisle and make sure that, you know, that thing's going to be going off all the time in Safeway when you're shopping for groceries. But what I do in everything that I do, I try to integrate those three things. How sick is that person likely to be? How close am I to that person? And how long have I been that close to that person? Okay. So I try to avoid situations where I'm going to be very close to people for a very long period of time, especially if there's going to be a lot of shouting, singing, or laughing, or, you know, or projecting. So what does that mean in practice? When I go to the store, I try to go to the store only when I've got a list, okay? I've got a list of things we absolutely have to have. I don't go every day. I try to go once or twice a week, or 3 times a week at the most. I go with a list. I don't take a shopping cart. I take a shopping basket, because I'm only trying to buy those things that I need to satisfy the next few days. I'm masked. And out in California, you can't get into a store unless everyone is masked. They won't let you in. That's not true everywhere. And then I zip around. I try to be in and out of that store in less than 10 minutes or 15 minutes. And part of that, of course, is avoiding the checkout line. So I'm one of those brave souls who goes to the auto checkout line and learn to master all the little tricks of checking out by myself. I'm in and out of there in less than 15 minutes. And my exposure to any one individual is probably minuscule. When I have to stand in line for the checkout, I am 6 feet away from the person in front of me. But I don't get paranoid if I'm 3 feet away from them, if we're both masked and it's going to be less than a few minutes. And I certainly don't get paranoid about crossing paths with somebody in an aisle if we're both masked. Now, if we're not masked, then I'm much more diligent about trying to stay 6 feet apart, because that 6 feet rule is designed to move most of the large droplets, have them fall to the ground before I'm in your effective radius. But the great virtue of wearing masks is you can get a little bit closer as long as those contacts are brief, right? So now, one other thing that I think is important is about contact with surfaces. There's a lot of stuff on the video about. My sister sent me a video that some guy made in the Midwest. This guy is virtually sterilizing everything that comes into his house. That's not necessary. The guidelines, yes, you can acquire infection by contaminated secretions being on a surface, and then you touch that surface and then touch your eye or your nose or your mouth. That's well documented for the common cold. But the common cold is an illness in which runny nose is a very dramatic part of the common cold. And large amounts of snot are pouring out of people, and especially children with common colds. SARS-CoV is not that. COVID, the runny nose is a very minor part of COVID. Coughing is the major part of COVID, that symptom of COVID that is involved in transmission. But COVID, asymptomatic carriers of COVID aren't producing gobs of snot and massively contaminating surfaces. So I don't decontaminate everything the mailman brings. I don't swab down everything that comes from Amazon.com to my house. I do quickly decontaminate stuff that I buy in the food store that is going into my refrigerator, okay? Because the low temperature of a refrigerator will allow virus to remain viable for longer than the ambient temperature of anything outside. So I wipe down with alcohol the milk bottle, the salad bag, stuff that's going into my refrigerator. Everything else, I just put away and wash my hands. I'm very diligent about hand washing. I'm diligent about mask wearing in public, even when I'm outdoors, but especially when I'm in an enclosed space. I try to keep my residence time in an enclosed space to very short, as brief as I can. But I don't go home and lose sleep over it if I'm there 20 minutes instead of 15. I just try to expedite it.
Great. Well, Don, this has been amazing, and I wish we could go on and on.
Yeah, that's the problem. I'm very hard to stop.
I have many questions myself, but folks here who emailed questions in, I think we should start getting to them. And then I know we have some Q&A on the chat. So here's a question from one of your classmates, Bing Chang, also class of 68, and he is the director of an Institute for Public Health and Medicine at Northwestern Medical School. And you touched upon this a little earlier, so he may want you to expand. He asks whether getting infected by a certain strain of the virus causing COVID-19 would likely convey immunity to other genetic strains, and what the implications of different genetic strains of the virus have for vaccine development.
Yeah. Okay. It's a great question. Being as a very well-known academic rheumatologist and a real expert about the immune system. So here's the story with SARS-CoV-2. Right now, we believe that all of the sequences that have been published are members of a single serogroup. There's a lot of individual mutations that accumulate in the viral genome as it spreads from person to person. Mm-hmm. Some of those have been posited to possibly affect spread, the efficiency of spread. Some of them definitely affect the stability of the envelope protein on the surface of the virus that would be predicted to affect spread. But none of them are so different from the parental sequence that we think they would constitute a different serotype, and they would not be protected by the antibodies that we're raising. So far, the good news is, first of all, the mutational rate of this virus is lower than most other RNA viruses, for reasons that I could explain to Bing, but would mystify the rest of you. But Bing, just between you and me, there's a 3' exonuclease in the genome that is a proofreading function. It's the only RNA virus that has that proofreading function. That's why the mutation rates are lower here. For the rest of you, all you need to know is that right now we believe that all the circulating SARS-CoV-2 isolates are of a single major strain, and that infection with one would be protective against infection with the other ones. That could change over time, but right now that's the view. There are mutations that have accumulated in the viral genome, and some of them may affect transmissibility. That's very controversial, but none of them would be expected to not neutralize the other ones.
That's great. That's good news. Here's another really interesting question from Susan King, class of 88. And she's asking on behalf of her kids who are meant to get vaccinations this summer. And doctors have been urging patients to come to their offices to get their routine vaccines. And she's wondering whether there are any thoughts for if, on what happens if children or young adults receive like a Tdap or Gardasil or even the flu shot eventually, when they could be asymptomatic carriers of SARS-CoV-2 or had been recently infected with SARS-CoV-2. Could that be harmful to them? Would the shots work? Would their immune system still develop antibodies to tetanus, and et cetera? Or are their immune systems so consumed with fighting off coronavirus that they wouldn't confer the same immunity?
Yeah, okay. I don't think there's any evidence yet that one should be concerned about this. In places that have been, where there's an obvious epidemic going on, when we do community-based surveillance, somewhere around 1 or 2% of asymptomatic people in those highly exposed populations are positive. So this is an uncommon event, okay, at the population level. It's common enough that these people play big roles in the silent spread of the infection in the community, but still on a per capita basis, they're not common. Okay, so the likelihood that your kids, especially if they've been domiciled with you for the last 3 months, are going to be positive is not very high. But even if they were, we would not discourage them from being vaccinated. Why? It's particularly important for them to get the flu shot and all the other measles shots and all the other things that kids get to prevent respiratory infection. Because in the next few months, just think what it's going to be like. Every time you get something that's associated with a cough, you're going to be wondering, is this a common cold? Is this the flu? Is this COVID? Is this RSV? Is this metapneumovirus? And so I think whatever we can do to prevent other respiratory infections from happening will simplify your life a great deal as a parent and their life as a student, because they're going to be wondering and they're going to be calling you up. Should I go to the infirmary? Should I, should I go to student health? Should I go to occupational health? Because I don't know if I might have COVID. Right now, if they're asymptomatic, they're not going to be screened before they go to be vaccinated. And we would say they should be vaccinated. And I think I double checked with my wife, who's the head of pediatric infectious disease at UCSF. She agrees with me very strongly. It's really important for these kids to be vaccinated, and we know of no reason not to vaccinate them and lots of reasons to vaccinate them. It is true that we encourage kids who are sick not to go get their vaccines. If you're sick with a common cold or with measles, it does impair your ability to make a good response to the vaccine. But that is not true for asymptomatic people who carry these viruses. We know that young children, 10 or 20% of them, are asymptomatic carriers of common cold viruses, and yet they respond very well to vaccination. We don't screen for that. I would treat this in exactly that way. If they're an asymptomatic carrier, concern about that should not keep you from taking them to the pediatrician and getting them vaccinated.
Great, thank you. Now, I have a very special series of questions here from a very special former student, Nanye Odukwe, and she is in med school right now. She said that she participated in a Columbia webinar yesterday with an alum who's the CEO of Regeneron. And she says that the IL-6 antibodies weren't effective, unfortunately.
Yeah, I've been hearing rumors about that. That does not surprise me.
On off the map.
Because many cytokines are up during that inflammatory state. And it was, and the antibody only neutralizes IL-6. So you were betting that was the dominant cytokine that was driving the process. And there are very few inflammatory processes that are driven only by IL-6. There are a few in multicentric Castleman's disease and in because you're a med student, I can talk to you this way. Multicentric Castleman's disease and the cytokine storm that follows CAR T-cell infusion, those are disproportionately influenced by IL-6, and IL-6 neutralization is very beneficial for them. But most other syndromes of inflammation are not dominated by IL-6 in that way and don't depend on it uniquely the way those do. So it doesn't surprise me that clinical trial failed.
So she has a follow-up question, which is more general about becoming a doctor. So she's a rising third year who's starting rotations in a month. And the general consensus among her classmates and many med students around the country is that they don't feel prepared to deal with another pandemic when they become in charge suddenly. And she wonders first how you would counsel them. And secondly, what do you think the nation should be or is perhaps doing to manage this second possible wave of COVID-19 in the upcoming flu season?
Yeah, so these are great questions. And she's not the only med student who's wondering about them. But as somebody who's lived through multiple pandemics and was, 2 influenza pandemics, and the entire AIDS epidemic, which went on for 20 years in my professional life, we had to confront this issue. When the AIDS epidemic began in 1981 and 82, I was a fellow in infectious disease. And, every night I went home, and San Francisco was the epicenter of the epidemic. And every night, the whole hospital was filled with AIDS patients by the time 85 and 86 rolled around. We had no therapies for anything. We understood very poorly how disease was transmitted. Needle sticks were common in the hospital. And, we went home every day wondering, was I bringing this home to my family? So you aren't the first generation to confront a potentially fatal infection that it's your responsibility to take care of. But this is our job as physicians. This is what we signed up for, knowingly or not. And the good news here is that with proper PPE and proper training, one can mitigate this risk. I think what made it particularly bad in New York, and especially in the Bronx and Brooklyn, was there was not adequate amount of PPE, and we had no idea, how overwhelmed we were going to get. Hopefully, the hospital administration has gotten that situation under better control, although the federal government bears a very heavy responsibility here and has really defaulted on this in a serious way. All I can tell you is this is our charge as physicians, and you're not the first generation to confront it. My father was a doctor who went to medical school in the 40s. At his time, tuberculosis was untreatable, and they took care of a lot of patients with tuberculosis. In my generation, it was HIV, and now it's COVID. The risks are significant, but they can be mitigated, and it's our job. And it's a hard message, but I think we can do a lot here to mitigate it. We need to keep our shifts a little shorter so that we're more alert. We need to have more frequent shift changes. We need to have an abundance of PPE so that we're not reusing masks or using substandard equipment the way we had to the first go around. So I'm hopeful that we'll be in a better place in terms of mitigation. I'm hopeful that the medicines we talked about will make, if you should get sick, there probably won't be any for you to take as an outpatient, but should you be hospitalized, there'll be a better standard of care. And you as a young person have a lower risk of a severe or frightening infection. But this is not an infection to be trifled with. Even the outpatient infection can be quite severe. And we're still learning about the prolonged recoveries of many people who've been ill. So I don't, I'm taking your issue seriously. I don't mean to be implying that there's nothing you can do. I think hospitals will be better prepared the next time around in terms of PPE and protocols and shorter shifts. But I think the core issue here is our moral obligation to take care of the sick. That does not change. That does not go away. That is something where, at the end of the day, we simply do have to accept that is our job. But hopefully, these other things will mitigate a lot of the concern. We know a lot more than we did when this began. We have better protective equipment, better routines. But at the end of the day, that uncertainty about whether you will be exposed and get sick, that is an intrinsic part of being a physician. And there isn't much I can say to make you feel better about that. That is what we do.
I have another question from a former student, Kevin Newhall, who's at Case Western Reserve getting his, doing a dual degree, MD, PhD. And he wants to know, he's curious about your perspective on the timeline for the pandemic. Will we be social distancing for years? What types of factors are, obviously vaccines and treatments, but any other factors that are timeline dependent?
Yeah, so we've talked about the treatments. I think not a lot of treatments are likely to come on the horizon before the flu hits in November and December. They're all in clinical trials now, which means that it's going to be a while before we know. In terms of vaccine, there's a lot of, a lot of magical thinking going on about vaccination. I've given you the good news, which is that so far animal experiments are suggesting that there is non-sterilizing potent immunity and that we can induce that with a vaccine. The other good news is there are 98 different vaccine preparations that are being rolled out simultaneously. So that's great. I mean, there's no other infectious disease for which that many different approaches have been tried. They're all variants of the same thing. They're all trying to induce immunity to the surface glycoprotein. But the good news is that if there are side effect profiles, they're likely to differ from one vaccine to another. And that by doing multiple of these, we can come down to a good, we'll have a good roster of options at the end of the day. The idea that we're going to have a vaccine by the end of this year, which the administration likes to talk about, that is lunacy and fantasy. Even if the clinical trial is done by the end of the year, upping the manufacturing and all that is going to require, a full calendar year from now. So we're all going to have to, I think, accept that that there won't be a vaccine for at least a year from now, and even that's pretty optimistic. Okay, now a true religious miracle can happen, and we can pray for it, but I'm not organizing my life around the idea that a vaccine will be available before at least a year from now, and perhaps longer. Okay, what does that mean? That means that we all are going to have to calibrate our activities around mask wearing and social distancing. What does that mean for me? As I'm retired now, so I don't have a regular job to go to, but I'm a very avid hockey player. I've been playing hockey twice a week for the last 20 years, and I have to give that up. I can't, even though the rink is now reopened and the state of California says it's okay to go there, I can't be on a bench shoulder to shoulder with 6 or 8 other people who are all exhaling very heavily the way I just showed you in that video. I just can't afford that because I turned 70 this year. I have to, I might have done that when I was 30, but at 70, I can't afford to take that risk. I'm going to have to stop playing hockey for a year. So this is a little bit of individual risk calculation. If something involves, if you're in a church choir or if you're in a choral group, I really think this is not the time to be in a choral group. This is not the time to go to a gym, okay, where people are exercising very hard and sweating profusely, and they're not coughing, but they're breathing very heavily. And that mist is being deposited on the surfaces of the gym equipment. I don't care how stringent the gym is about wiping everything down. They're never going to get every last particle off that machine. And so I'm not going to the gym either in this time, which is killing me. So I have to adjust I bought a treadmill machine for what used to be my kids' playroom. I'm on that 45 minutes or an hour a day, but I'm not going to a public gym. Okay. And I probably won't until there's a vaccine. Now, if you're younger and have more risk tolerance and you're at a lower risk for severe illness, you might make that calculation differently. But these are the sorts of things that I'm thinking about now. I think we should count on a calendar year before there's even a remote chance of having a vaccine available for you, and thinking about getting into that psychic frame of mind in terms of your work life and your recreational activities, that social distancing, mask wearing, hand washing, this is going to be with us for a long time, and calculate accordingly.
So there's a question about viral loading. You talked a lot about the large droplets falling to the ground or falling onto the surface in a gym environment. What about the smaller droplets? Do those accumulate within the room if there's not good ventilation? What is your thinking on that?
So, this is a really difficult question because we don't have chapter and verse in a way that would satisfy a rigorous scientist. When I was in medical school, we were taught there were 2 kinds of respiratory spread. Droplet spread, with these great big droplets that fall to the ground within 6 feet, and then what was called aerosolized transmission, in which very small droplets can hover in the air and kick around with by Brownian motion for a long time, hours, and that diseases were felt to be in either one of those or the other category. That measles and chickenpox are examples of aerosolized spread, where just coming into the same room with a kid with chickenpox for a very brief period of time can lead to a significant exposure. In chickenpox, it's so contagious that if one member of your family remembers having had chickenpox, you can count on the entire family being seropositive, having been exposed. Same thing with measles. If one kid in the family had measles, everybody got exposed and is going to be considered to have been infected. Whereas for SARS-CoV-2 in the household, it's something like 10% of the household contacts, depending on the household, may get infected. In a common cold or influenza, maybe 20% or 30%, depending on the age of the household and how closely they are together. There's no absolute, this thing the way we were taught about this in medical school, which is made it into textbooks and now onto the internet, is this binary distinction. There are infections that are like that. The common cold at one end and measles at the other end. And, but SARS-CoV-2 is more likely to be an intermediate case where the predominant route is large droplets within 6 feet, okay? But for people with very high titers, the so-called super spreaders, they might be able to produce enough virus in those tiny little droplets that go out into the atmosphere to occasionally transmit to somebody. We don't know that. This is an unknown, okay? So what I would say is, as a practical matter, is this, that we should assume that there's a continuum of particle sizes between the big ones and the small ones, and that especially if you're going to be in an environment where people are breathing very heavily, they're going to be generating very large numbers of both kinds of droplets. And for the most part, it's going to be the large droplets that do most of the transmission, but in the rare person with very high titers, they might be able to contaminate those small droplets. You have to be concerned about that. That's why indoor events are inherently more risky than outdoor events, where the wind is carrying them away to effectively infinite dilution in the sky. Right? So I can't answer your question with a great deal of rigor. All I'm saying is that you have to think about risk semi-quantitatively, understand what we know and don't know, and therefore closed spaces are worse than open spaces. Heavy breathing, singing, vocalizing, shouting is worse than simple speech, which is worse than utter silence. And that for the most part, most spread we think is accountable for by large droplets. Why do I say that? I say that because the infective rate, the R naught for spread for diseases like measles and chickenpox is like 15 or 12 or 18, whereas the R naught for droplet spread is typically 1 to 3. You know, and R naught for most of the epidemic of COVID is debated between 2 and 4. So it's closer to that, which is characteristic of droplet spread. So for the most part, we're talking about large droplets here. I can't say there isn't an occasional transmission by that airborne route, but we can't organize our entire public health practice around that rare event. We organize it around the common event, and we should organize our individual personal lives around those common events. So think about it the way I just said. Stay away from things that induce heavy breathing in a closed space. I mean, to me, a gym, a small retail gym in a strip mall is the worst place you could be right now, probably. A choir group, a choral group in a church is not a bad place. Any big environment that's in a closed space, the mega church, the political rally, is worse. These outdoor rallies are better than indoor rallies, but they are not entirely safe. And I think we should be honest with ourselves, even as much as we are sympathetic to the current movement in the streets. I certainly am. I can't say that that's an entirely safe activity. It's not, even with all the mask wearing, because I'm still seeing about 1 in every 3 or 4 people is not wearing a mask. And the riot police, interestingly, with their shields, are typically not wearing masks because I think they think they're being protected by their shield. But there have already been a half a dozen cases among the riot police. So that tells you that even the dilution factor, when you have that many people crowded for that long, you know, it's still a risk. And I remind people that in 1918 in Philadelphia, they had a victory bond march that went on all day on Main Street in Philadelphia. And that was a big amplifier of the epidemic in Philadelphia. So these outdoor events cannot be considered to be entirely safe. They are safer than an indoor event, but they are not entirely safe.
Great. I think we have to stop with one final question, super practical, from Dr. Ramos, who is the chair of our history department and joined us tonight. She asks, what type of masks are most effective for daily use?
Yeah, okay. So we'll close with this. The typical mask that people use for this kind of routine droplet control, and which you saw in that video, is a surgical mask or something that mimics a surgical mask. The kind of masks that you make out of a bandana are not as good, but they are better than nothing. And when surgical masks were in a great shortage, when there was a great shortage of them, and you couldn't get them on Amazon, a cloth mask is fine. It's perfectly good for most things. I feel very secure going into Safeway when other people are wearing a cloth mask. I happen to have a box of surgical masks that I got on Amazon after a number of weeks. I'm going through that right now. So those paper masks are made, they have 3 layers of paper in them. And there's at least a lot of standardization that goes into the construction of those masks, so that that they are probably slightly more efficacious than a cloth mask. But if you can't get them, the cloth masks are more than adequate for most of this, of what you need. And the other thing about the cloth masks are they're much more economical because you can reuse them. You can wash them and reuse them. A paper mask, once it gets moist, is no longer as effective as a barrier. Okay. Once those masks get wet, they really lose a lot of their filtration properties and their ability to retard more droplets. So if you wear a mask like that for more than a few hours, you'll see that they start to get moist. That's an indication of how well they're functioning, right? And the skin around your chin is going to get moist. That is an indication of how well they're working. But it also means the moister that mask gets, the less effective it's going to be. So you really shouldn't wear an individual mask for more than a total of 6 or 8 hours. Then they start to get so wet they don't filter well. Now then you have to throw them out and get another one, which at $1 a mask really gets to be very expensive. Whereas your cloth mask can go right into the washing machine and come out and be dried and be ready to use again. So I'm perfectly comfortable with cloth masks, or the paper, sort of ersatz surgical masks that clip on behind your ear. Either one should be okay. But you should be aware that if you use these paper masks that are slightly more efficacious, just slightly more, they do decay when they get moist, and you have to throw them away. You can't dry them out and use them again.
I know a lot of the masks have these inserts, the charcoal inserts or various filtration. How much does that actually help?
I don't know the answer to that question. When you go on the internet to buy a cloth mask, they'll say that they have a pocket for you to put the insert in. When I looked up what these inserts are, they're mostly little bits of activated charcoal, little things of activated charcoal. These were designed for industrial purposes. Activated charcoal is there to absorb environmental soot and stuff like that. So for industrial workers, that's why these masks that's why you can get those inserts. How effective they are at retaining your own secretions, I really can't say. I'm sure they don't hurt. Do they help at all? I don't know. And I'm not sure I know anyone who does. It's not something that's been studied carefully. But those were designed for industrial purposes, for filtration of soot and airborne particulates, not for the trapping of your secretions, which is what we're trying to do here.
Great. Thank you so much, Don. This was amazing. And we're so lucky at Andover to have you. You've been such an incredible alum.
Ask me back. I love doing this.
Oh, we will.
Yeah.
Okay. Thank you.
Thanks, everybody.